So we're going to first welcome Melissa Bateson. Professor Bateson is the director of Newcastle University's Center for Behavior and Evolution. As an ethologist, her research focuses on understanding how stressful lifetime experiences impact behavior, cognition, and well-being of various non-human animal species. Welcome, Professor Bateson.
Thank you, Anne, for that introduction. Can everybody hear me okay?
Yes.
I'm gonna, and also I need to share my slides. Okay, can people see my slides now?
Yes, I can see.
Okay, great. Okay. Sorry, let me just. Oh, sorry, I'm just trying to. Okay, so I need to thank the Wild Animal Initiative for inviting me to give this talk today about validation of welfare indicators. It's something that I've been thinking about a lot just recently. I've been contributing to a book on welfare indicators that's being edited by Georgia Mason, Mike Mendel, and Berta Nielsen from U4. I'm contributing one chapter in that book on telomere length as a welfare indicator. And one of the things that all of the contributing authors have been asked to do by the editors in this book is to really think about how valid our indicators are. So it's something that a group of us have been discussing for a while now. And what I'm sharing with you is a kind of summary of the basic methods that Georgia has encouraged us to think about in writing our chapters for this book. So I hope that what I say today maps really well onto what's going to be in this upcoming book, and that many of the people here will be able to benefit from that book in the future. It's due to be published next year, I believe. Okay, right, so let me go on to the next slide. Okay. So I just wanted to start by asking what is animal welfare, because there are many different definitions and conceptions of what we mean by animal welfare that exist in the literature. And I wanted to focus on this particular one, this 5 domains model from Meller. I like this model of animal welfare because it acknowledges that many different domains feed into animal welfare, but it puts the mental domain at the center here. So although it acknowledges that nutrition, the physical environment, health, disease and injury, and the social environment and opportunities for behavior all affect animal welfare, the idea is that these are channeled through the mental effects on animals. So if we focus on emotional or affective states which might arise as a result of these various functional domains, that gives us the best way of thinking about what we mean by welfare. And I think the reason for this is that it's the worry that animals might experience negative emotions such as thirst, hunger, anxiety, fear, pain, distress, these unpleasant negative emotions that really drives our concerns about the welfare of animals. And also the possibilities that they might be able to experience positive states such as excitement or satisfaction, relaxation. Positive states are important too. But the nice thing about this model is that it puts emotional or affective state at the center of what we care about when we talk about animal welfare. Okay, so a problem we face here is that in ethology, subjective experiences in animals were not really regarded as a proper subject for scientific inquiry until relatively recently. So I put up this quote from Nico Tinbergen here. He says that because subjective phenomena cannot be observed objectively in animals, it's idle to either claim or deny their existence. So Tinbergen really thought we shouldn't be talking about mental states in animals, and that the whole domain of what we think of as being central to animal welfare now was not something that could be addressed scientifically. So I think things have changed a lot since Tinbergen wrote this, and now I think especially as animal sentience is now recognised in, for example, UK law, we have the Animal Welfare Sentience Act, which acknowledges that vertebrate animals, decapod, crustaceans and cephalopods are sentient, by which we mean that they're capable of experiencing feelings that might be positive or negative. We really actually need to talk about how we can measure these things objectively, and that's what I'm going to be focusing on in my talk today. So the trouble is that there are lots of problems with inferring mental or affective states in animals. We don't know which species or stages of development have affective states. So I just mentioned our UK Animal Welfare Sentience Act, which makes some decisions about this, but people might agree with those, evidence might change. We don't know at what stage in development animals become sentient. So there are a lot of question marks there. Also, we have to acknowledge that animals' affective states might be very different from our own. We just don't know the answer to this question. And the general problem we're facing here is the other minds problem, the fact that subjective feelings, even in humans, are private and inaccessible. I mean, we at least can tell other people how we feel, but even then we can't know for sure what it's like for another person to experience a particular emotional state. So when we're trying to assess affective states in animals, we need objectively measurable indicators of affective states. And we have to acknowledge that we can really only assess these. We can't directly measure them because we don't have any access to them. Okay. So I want to talk a bit about the characteristics of a good welfare indicator, a good indicator of affective state. And I'm mainly going to be talking about validity in this talk. So an indicator will be valid if it's measuring the right thing. Indicators also need to be reliable, which means they need to be repeatable. So if we do a study again, we need to have repeatable measures. So I just wanted to start, before I go into validity in more detail, talking a little bit about accuracy and precision in measurement. So both validity and reliability of indicators are affected by accuracy and precision of individual measurements, but there's no one-to-one mapping between accuracy and validity or between repeatability and precision. And I wanted to say this because I think a lot of people think that validity and accuracy are the same thing, or that the precision of measurements directly maps onto reliability. And I don't think that either of those things is true. So I just want to clarify that before I move on to talk more in detail about validity. So let's just think a little bit about accuracy versus precision to start with. So what I want to make clear here is that these are attributes of repeated measurements of the same underlying quantity. So in this figure here, the bullseye in these targets represents the true quantity we're trying to measure, and the black spots represent independent attempts to measure that quantity, so measurements we've actually made. Precision, which is the vertical y-axis in this matrix, represents how clustered our points are. So you can see that in the top row, our spots are very close to each other in both cases, whereas in the bottom row, our spots are much more spread out. So we've got high precision of measurement at the top here and much lower precision at the bottom. Our other axis here is accuracy, which relates to how close the mean of our points is to the true quantity we're trying to measure, which is at the bullseye. And you can see that on the left, we've got poor accuracy. So the mean of the cloud of points here is not going to be very close to the bullseye of the figure. Whereas on the right here, if we take the mean of the cloud of points, it will be approximately on the bullseye. So accuracy relates to whether our measurements are biased, as would be the case on the left, or whether they're accurately assessing the underlying quantity we're trying to measure. So I think it's true to say that accuracy and precision of measurements affect validity and reliability of indicators, but they're not the same thing. And I hope to try and explain that in the next few slides. Accuracy and precision are attributes of repeated measurements of the same thing, but they're not the same as validity and reliability. So I think I'll get reliability out of the way first. So reliability is about repeatability, the extent to which measurements are repeated when a study's done under the same conditions. Unreliable indicators will usually have high measurement error, and both the accuracy and precision with which individual measurements are made affects reliability. Reliability will be higher if measurements are more accurate and more precise. What about validity? So validity concerns the extent to which an indicator measures what we actually want to measure. In this case, affective state or subjective feelings of an animal. So this type of validity is sometimes called construct validity. In psychology, you'll find many different kinds of validity referred to, which can be very confusing, but construct validity refers to what it is we're actually trying to measure here. And I think in general, validity is much more important than reliability. It's better to measure the right indicator somewhat unreliably than the wrong indicator very reliably. And I think you can see why that would be the case. Unlike accuracy, validity is not an intrinsic property of an indicator, but accuracy is an important component of validity. An inaccurate or a biased measurement will not generate a valid assessment of affective state because it's just measuring the wrong thing. It's off target. Validity has to be established by comparing the indicator with a gold standard or ground truth. So this is what I'm going to spend the rest of my talk talking about, how we can actually do this and what the methods are available to us. But the question is, how can we do this when the feelings of animals are inaccessible to us? I've already told you that since subjective feelings are private, we can't get at them. So how in the world can we go about validating any indicator of welfare if the gold standard or ground truth is not accessible? So that's the million-dollar question we need to try and address here. So the solution to this is to realize that subjective feelings are only one component of affective state. They may be the component that we're most interested in when we talk about welfare, but most affective states have other components to them as well. And these include cognitive components to do with how the animals process information, physiological components, which might be to do with, for example, hormone levels, and behavioral components. So I've shown an example here for hunger, which is something that a lot of my research relates to. The subjective feelings that we're interested in relation to hunger are going to be things like fatigue, hunger pains, anxiety, distress, and possibly also anger and shame. So in humans, these are all emotions that we know go with being hungry in one way or another when hunger becomes distressing. However, we also know from many studies that when people have these subjective feelings, that goes along with changes in cognition that we can measure objectively. So, for example, we can measure changes in the value that humans or animals assign to food, their attention to food, how impulsive they are, whether they prefer to get a small food item sooner rather than a large one later. So these are all aspects of cognition that we can measure objectively in an animal. Similarly, if we move on to physiology, there are things that we can measure that might relate to hunger. We can measure, for example, blood glucose, which might be very low in an animal that's chronically hungry. We can look at appetite hormones such as leptin and ghrelin. And we can look at stress hormones such as cortisol that might be higher in a chronically hungry animal. Similarly, we can look at behavior. So we can look at time spent foraging, speed of eating, food preferences. So perhaps tolerance of eating lower quality foods or foods containing toxins. We can look at risk proneness. We can look at aggression. So these are all behavioral variables that might be good indicators of underlying hunger. So the idea here is that physiology, cognition, and behavior can all be measured objectively in animals. The thing we might be wanting to get at is the mental domain, the subjective feelings. But what we have to realize is that we can only really infer subjective feelings from measurements of valid indicators. And these are probably going to come from the domains of cognition, physiology, or behavior. So the problem we're facing is how to figure out which of these many things that I've got under cognition, physiology, and behavior might give us the most valid assessments of this domain of subjective feelings. Okay. So I'm going to talk about 5 different methods that we've identified for validating indicators of affective state in animals, which I'm labeling 1 to 5, which is also the same scheme that Georgia Mason is using in her book, I believe. So this should map on to the scheme that she is proposing for how to do this. Briefly, the 5 methods are, method 1 is assessing whether the indicator tracks self-reported affective states in humans, and I'll talk about each of these in more detail in a minute. The second is assessing whether the indicator changes in animals exposed to preferred or aversive conditions. The third is assessing whether the indicator changes in animals exposed to fitness boosting or harming conditions. The fourth is assessing whether the indicator changes in animals given affect modulating drugs. And the fifth is assessing whether the indicator co-varies with pre-validated indicators of affective state. So I'll say a little more about each of these now. So method 1. So this is the idea that we can look at whether the indicator that we're trying to validate co-varies with self-report of positive or negative feelings in humans. So the reason for using humans here as our gold standard is that humans are the only species that can actually tell us how they feel in different conditions. The argument here from a homology is that if affective states have similar correlates across species, then we can assume that if a human is reporting negative or positive feelings in association with an affective state, then we can assume that animals would also experience those same feelings, even if they can't report them. So in a way, this is perhaps a sort of gold standard, because we're actually using subjective feelings here to validate an indicator in humans, and we can then take that indicator and use it in animals and assume that it will be a good reflection of the feelings of that animal. So I've worked a lot with telomere length, which is a cellular biomarker of cumulative exposure to negative affect. And one of the reasons for focusing on telomere length is that there's a huge amount of data from humans showing that shorter telomeres are associated with states in which humans report feeling miserable in one way or another. So, for example, humans with clinical depression have shorter telomeres. Humans who are anxious have shorter telomeres. And in general, humans who report more subjective stress in their lives have shorter telomeres. So this huge body of data, and there are meta-analyses of all of these things in humans in existence, really convince us that in humans at least there is this association between telomere length and negative subjective feelings. And also, actually, on the other side, there are also some studies suggesting that longer telomeres might be associated with positive feelings in humans. So this is a very sort of powerful way of validating a cellular biomarker that we can also measure in non-human animals that can't tell us directly about their feelings. So as I've said, this validation approach is based on an assumption of homology, that affective states have similar correlates across species. The validation is stronger if affective state has been experimentally manipulated in humans. So if we know, for example, that the indicator we're interested in this case, telomere length, changes when we experimentally change affective state in humans, then that's a stronger validation than just showing that there's a correlation in observational data. One of the problems with telomere length is that the states associated with shorter telomeres in humans are not things that it would be ethical to manipulate. So we can't experimentally make people depressed or anxious just to validate a biomarker. But interestingly, we can make them happier and see whether you get the opposite effects on the biomarker. So for telomeres, there are actually studies suggesting that making people partake in meditation, for example, actually leads to positive changes in telomere length. So for positive affective states, it may be possible to actually have experimental evidence from humans showing that when you change the affective state, you also change the indicator in the expected direction. So this method one, unfortunately, breaks down when indicators are unique to a species. So telomeres are good because all eukaryotic cells have telomeres, so they're quite a widespread indicator. But for some indicators that we might want to use, like, for example, crying in humans, isn't routinely seen in other species. So it's hard to use this approach. So it's not going to work if the indicator of interest that we're trying to validate isn't something that is present in many different species. Okay, so moving on to method 2. Okay, so method 2 is a method that we can use in the animals themselves. It doesn't rely on humans. And the idea here is that a valid indicator should co-vary with exposure to conditions that the animal would seek out or actively avoid if given the choice. So if the indicator is an indicator of negative welfare, it should co-vary with things the animal should actively avoid, or if it's a positive indicator, it should co-vary with things they should actually seek out or try to expose themselves to. So, for example, we can think of things like indicators that change when a rat is given an enriched cage, which is something which we could show that a rat might seek out. If a polar bear is confined to a small enclosure, a monkey's socially isolated in a cage, a broiler chicken is housed on wet, dirty bedding, etc. So we can think of many kinds of studies we could do to show that indicators change when we change the conditions of an animal in a way that we know that they would either avoid or seek out. So this approach to validation assumes that positive and negative affective states have evolved to promote approach in the case of positive ones and avoidance in the case of negative ones. This is the assumption about why we have emotions. They are there to motivate adaptive behavior. Positive emotions make us seek out things. Negative emotions make us avoid them, move away from them. This kind of validation is stronger if the conditions have been experimentally manipulated in the animals of interest. So, for example, it would be really useful if you could show that when you experimentally change the amount of enrichment a rat has in its cage, the welfare indicator that you're trying to validate changes in the predictable way with that manipulation. So, for example, if you think that pacing might be an indicator of poor welfare in a polar bear confined in a small pen, then you'd really want to show that behavioral indicator would change when you change the environment of the animal. With this method of validation, difficulties can occur when the conditions are new stimuli to which the animal has not evolved appropriate responses. So I think issues can occur with things like drugs of abuse or refined sugar. So these are things that animals' behavior has not evolved to cope with. And I think in these situations, there may be a conflict between short-term hedonism, so animals really wanting to access drugs or very sweet foods, and the longer term health and well-being of the animals. So it may well be that there can be issues here to do with conflicts between in what animals actually want in the short term and what's good for their health in the long term. And of course, health isn't necessarily the same thing as positive affective state. But going back to Mellor's model that I started off with, I think it's very often the case that poor health will actually lead to negative affect. So we do need to think about these things carefully. Okay. So going on to method three of validation is really very similar to method two. So the idea here is that a valid indicator should co-vary with exposure to conditions that improve or reduce Darwinian fitness in the species of interest. And I think here examples might be indicators that change when an animal is sick or injured. So the rationale here is very similar to with method two, but this method is particularly useful for situations where we can't conduct choice experiments for ethical reasons, such as in the case of disease or injury, where we may not want to actually do these things to an animal to validate our indicator, but we have a very clear a priori belief that that these kinds of things would be bad for the animal, bad for its fitness, and so the animals, for that reason, should be motivated to avoid them. So again, like with method two, the underlying assumption here is that affective states have evolved to promote fitness, so better survival and reproduction, and they do this by motivating animals to approach things that will improve their fitness and avoid things that will reduce it. Going on to method four, this is another method that we can use in animals, and the idea here is that a good valid indicator will be affected by drugs that alter affective state directly. So this is often known as pharmacological validation, and I think for some people it's regarded as one of the kind of gold standard ways of validating indicators of affective state in animals. So examples here would be behaviors that reduce when an animal that has just had surgery is given an analgesic drug that kills pain. So this would be a way of validating indicators. Validating indicators of pain. These would be behaviors that change when you remove the pain from the animal using a drug that you know does that in human subjects. So usually our choice of drugs is determined from the effects that they have in humans, and then we assume that those drugs will have the same effects in animals and that we can use them to validate behavioral indicators of negative affect in animals. So it relies heavily on this assumption that drugs have similar affective states in different species. Okay, and the final method I want to mention, method 5, is that indicators can be validated if they co-vary with other already validated welfare indicators. So clearly, if you have a validated welfare indicator in existence, then you might be able to validate a new one by showing that it validates with that existing one that will have had to have been validated by one of the earlier indicators. So we can use indicators to validate each other to some extent, although we have to believe that we're building on sound foundations here, or we could just collect a whole lot of indicators that are invalid if we're not very careful with this approach. But it is an approach that's often used to say, look, I've got a new indicator here and it correlates with other things that I really believe are good indicators. Okay, so those are the 5 methods. Valid indicators also need to be sensitive, by which I mean good at detecting cases of negative or positive affective state, i.e. Avoiding false negative errors or what we might call misses, and I'll say a bit more about this in a second. They also need to be quite specific, and by that I mean not be sensitive to factors other than affective state, so prone to false positive errors or false alarms. Okay. So just to make this a little clearer, if we think about a group of birds, the birds on the left of this diagram are birds that have poor welfare. The birds on the right are birds with good welfare. We've got some indicator of affective state. In this case, an indicator that we think indicates animals that have poor welfare, and they're the ones. The animals with this indicator are the ones enclosed in the grey dotted circle here. You can see that we've got some errors here. So we've got, on the left, we've got false negatives. So these are birds that have poor welfare, but that have been missed by our indicator. So the green birds are the true positives, the ones that the indicator is picking up as having poor welfare. The false negatives are the misses who this indicator is missing to detect. Similarly, on the other side, we've got false positives, which are birds that are picked up by our indicator that actually have good welfare. So the indicator is suggesting they've got poor welfare, but in fact their welfare is good. So these are false alarms, or they're mistakes that affect the specificity of our indicator. The true negatives are the ones we actually want to have lots of, which are these grey ones on the right. So if we think about an indicator with poor sensitivity, might look like this, where we have lots and lots of false negatives. It's got poor sensitivity because it's not picking up a very high proportion of the birds that have truly got poor welfare. It's actually missing a whole lot of them. Whereas an indicator with poor specificity would look like this. It would be picking up a lot of birds and saying that they have poor welfare, when in fact their welfare is good. So something else is causing those birds to trigger that indicator. So it's not very specific. It's not causing us to really identify very well which birds have poor welfare. Another way of thinking about different welfare indicators is to think about this plot here, which Georgia has been encouraging people to plot for their indicators. So in this plot here, we can plot the valence of an indicator on the x-axis. So very negative states are on the left-hand side, very positive states are on the right-hand side, and then the value of our indicator is on the vertical axis. So for example, for telomere length, we might have short telomeres at the bottom and long telomeres at the top. Now, an example of an ideal welfare indicator will be one where there's a monotonic relationship between the valence of affective state and the value of the indicator. So the shorter your telomeres are, the more negative your affective state is. The longer they are, the more positive your affective state is. And also where we've got very few false negative errors, which are the errors in this quadrant, and very few false positive errors down here. So that would be a perfect indicator. And I think it's fair to say we don't have any that look like that. Many of them do part of this well, but very few indicators do all of this. Because of this, we really need triangulation. So none of the methods are perfect. They all rest on assumptions that might be flawed in specific cases. Therefore, what triangulation does is give us converging evidence from two or more methods that will strengthen the case for the validity of an indicator. So Pat Bateson said this in his paper about assessing pain in animals. He said, no single criterion provides an all or none test for the existence of subjective sense of pain. So he was very much in favour of this idea of triangulation, the idea that you need to not just rely on a single indicator, but that you'll have a much better idea that you're actually making valid measurements if you use several different indicators and use converging evidence for poor or good welfare in a particular case. Okay. So I just wanted to take one example here. There's often an assumption that body weight or condition is a good welfare indicator, and the assumption is that fatter equals healthier equals happier. And I just put a quote here from a relatively recent paper. Wild-caught animals may suffer from stress in captivity and lose weight, and environmental stress in poultry may reduce food intake and thus impair weight gain and reduce fat reserves. So there's this idea that body fat is a good indicator of welfare in animals. And it's an important, this is an important issue because weight is something we can very easily measure in wild animals. So I think it's a sort of simple indicator. We really need to know how valid it is. This is a picture that I drew recently. So this is George's graph of valence of affective state against body weight. And I think the problem with body weight is that while it is the case that very low body weight is associated with poor welfare, so we get the slope we want in this quadrant here. So definitely welfare goes down as body weight goes down at some point. But it's also possible that very high body weight is associated with poor welfare. Obesity might lead to poor health. Plus, we also know that animals tend to put on weight when they're food insecure. So certain sorts of unpredictability in the environment, like food insecurity, can lead to weight gain. So this is a case where both high and low measures can be associated with negative welfare. And there also might be a very high portion in the middle of this plot where there's a wide range of weights with very similar welfare, which means it's got quite poor sensitivity for picking up differences. So this would be quite a poor welfare indicator which is not very non-specific and also not very sensitive. Another one like this might be sleep duration. We know that depressed people sleep either more or less. So there can be a lot of indicators that have this quality that you need to be very careful with. Here's another one, acute glucocorticoid release might be an example of a very sensitive but not very specific welfare indicator. So cort might increase with very negative affective state, as shown on the left-hand side here, which is what we might want to see in a good valid indicator. But unfortunately, cort can also go up with very positive affective state. So when you're excited, for good reasons, you can also have high cort levels. High cort can be indicative of both highly positive or highly negative states. And this probably is due to the fact that it's reflecting arousal more than the valence of affective state. Okay. So just to summarize what I've been saying here, we need reliable and valid indicators that can be objectively measured to infer the welfare of non-human animals. Validity means measuring the right thing. We need evidence that the indicator that we're measuring co-varies with the valence of affective state. So the indicator needs to be telling us something about whether animals are in a positive or a negative state. I've suggested at least 5 methods that are available for establishing validity. But I think that no single indicator will be perfect. They've all got problems with sensitivity or specificity. Some indicators will have poor sensitivity, others will have poor specificity. And for this reason, we need to use a triangulation approach via 2 or more indicators. And this is crucial to increase our confidence in welfare assessments. So that sort of, I think, summarizes everything I really want to say here. And I just need to thank various people who've kind of contributed to this talk. I need to thank Paul Martin, who was my co-author on the 4th edition of Measuring Behaviour, with whom I spend a lot of time talking about accuracy, precision, reliability, and validity, and trying to sort those out. So I would encourage you to go and read our chapter on data quality in that book if you're interested in these issues. But I also especially need to thank Georgia, who's a wonderful friend and colleague, and who's challenged me to think very hard about validation of welfare indicators over the past couple of years. And as I say, many of the ideas in this presentation come from Georgia, so you should look out for her upcoming book on valid welfare indicators, co-edited with Mike Mendel from Bristol and Berta Nielsen from U4. And I'd finally just like to thank the Wild Animal Initiative for some current funding and for inviting me to share this with you today. Thanks very much. I'm happy to take any questions on that.